So now let's move on to Maurizio's presentation. His presentation will gonna be about clinical research and also in vivo experiment.
So thank you to JISS to accept my application and have me here. I'm also very proud and happy to bring neuropsychology up here. And I want to thank my supervisor, Anders, and Susanna to get me from the beginning. We are together from almost 2 years now. The data that I'm going to show are preliminary, and some others have been published. And I try to give you an overview. So as Take already introduced me before, I have 2 clinical studies and 1 preclinical study. The aim is the cerebellum, and I choose 2 specific disorder and 1 neurological disease where the cerebellum is clearly impaired. But at the same time, to make this study translational, I'm going to use NeuroPixel to record cell population from cerebellar cortex. And at the beginning, the cerebellum was known for motor function, motor control, vestibulo-ocular reflex, and sensorimotor integration. But thanks to neuroimaging and neurocognitive assessment, we have studies that show the cerebellum is active and contributes to higher cognitive function, like episodic memory, visuospatial ability, visual perception, and social cognition. And many others are not clear yet. So there require a lot of research. And we know that we have a neural pathway to cognition, so for example, to the prefrontal cortex or posterior parietal cortex and basal ganglia. So the first study is in neurophysiology, and we're going to use NeuroPixel. So if we start with the first image here, at the beginning we could register only 1 single cell. And then, thanks to technology, we could develop multi-unit recording, so multiple cell. And 4 or 5 years ago, more or less, we had the NeuroPixel, where on the probe we have 385 sensors, so we can record a cell population and not a single cell. The goal is to try to quantify and understand how is how cerebellar neurons during motor learning and skill acquisition. And here there are some pictures, some neuroimaging that show where the cerebellum is clearly atrophied in spinocerebellar ataxia. We have 48 mutations, but I choose spinocerebellar ataxia type 3, which is the most common in Sweden and in other countries in the world. And here we have gray matter reduction for ADHD and for autism. Yeah, so I'm going to present some results from the study in neuropsychiatry. And the idea is to solve the problem with neurocognitive assessment when biomarkers are missing. So for the neurodegenerative disease, we have biomarkers and usually help to identify the diagnosis. But for autism and ADHD, there are no biomarkers. So I required some objective methods. Often psychiatrists choose the wrong diagnosis, and it plays a huge role on patients because for ADHD, we have pharmacological treatment, but not for autism. So sometimes people don't get any diagnosis at all. We have 6 cognitive domains: attention, visuospatial abilities, social cognition, language, memory, and executive function. The idea is to create cognitive phenotype in order to differentiate, but with the contribution of neuromotor test. Neuropsychology started with this one of those first study, for example, to differentiate Alzheimer, then depression, when biomarkers are not clear yet. Then neuropsychology evolved a little bit more, and we could differentiate, for example, Alzheimer, then frontotemporal dementia, where there are no biomarkers. We know the neurofilaments are higher. Then progressive primary aphasia. We cannot choose any diagnosis, but we can help doctors to differentiate. So here are some results of a study that we published, did on children. And there are just two methods that I'm going to show. The first one is prism adaptation, and the second one is finger tapping. So we can start with this. Here there are the differences on the ability to maintain rhythm. And this is a pure cerebellar test where we have a clearly contribution thanks to neuroimaging studies. And patients that have ADHD have difficulties to maintain rhythm. And you can also see it with your own eyes here. And there we have a prism adaptation, which is another neuromotor task where patients need to use those glasses for 3 times. So 10 trials without glasses, 10 trials with glasses, and 10 without. And they have to point to a specific point. And the difficulties emerge when they need to find the right visuospatial relation. So here you can see that children without ADHD have less difficulties than those that have ADHD. And they have it during adaptation and after adaptation. And another study that is ongoing, but those are just preliminary results. We choose 2 groups. One group with people that have only autism, another group only with ADHD. Medicated. So now we are going to include in new analysis a group with ADHD non-medicated to see if they can have differences on processing speed. And we found that there are clear, I'm not going to explain every single test, but here we found clear differences between ADHD and autism than healthy controls. So they display a light sensory motor deficit, and those contribute to visuospatial ability. How do we measure that? Because it's also difficult to do this in real life with patients. We have some figures when they have to copy, and then we're going to correct the figure according to the rotation, the centimeters from the side, or if the line are in the same proportion. And we have seen, we found a good result at the moment. What we're going to expect is that, so if you look at the figure F, when we're going to check how is the completion time, we can see that ADHD group is slower than autism, and those are medicated. So we expect that patients with ADHD will perform even slower than those ADHD medicated, which can be an indicator to differentiate ADHD and autism about this cognitive domain. And the idea is to combine test result and the way of correcting tests with artificial intelligence and deep learning machine in order to have even a more objective way of correcting the test. So to conclude, we have seen that finger tapping tests and Prisma can predict and identify ADHD in children. The 2 groups of patients that I choose display sensorimotor deficit. And we have a clear cognitive impairment in the visuospatial domain, because when it's more than 1 standard deviation, it's an impairment, it's not a deficit. And when we don't have biomarkers, neurocognitive data can help and differentiate. And all those tests that I choose, this is only just a part. They can be used in clinical settings. And I wanted to show with this result that cerebellum contributes to cognition and not only to motor function. And that's it from me. Thank you.